Welcome to the Huberman Lab podcast, where we discuss science and science-based tools for everyday life.
Welcome to the Huberman Lab podcast, where we will discuss science and science-based tools for everyday life. Today I'm joined by Danielle Hancock, a professor of anatomy and sensory neuroscience, whose work focuses on how we detect, interpret, and respond to the world around us. We're covering general senses like taste, smell, hearing, balance, and vision, or essentially how your nervous system builds your reality.
Thank you for having me, Andrew. I'm excited because these sensory systems are the foundation of how we interact with the world around us, and most people don't realize how complex that is, even with a simple sensation.
Let's start broad. What are known as general senses, these include things like touch, pressure, temperature, proprioception, things we detect everywhere in the body. One thing people don't realize is that sensory receptors transduce energy, meaning they convert some physical stimulus, such as light, vibration, or chemical molecules, into electrical activity the nervous system can understand.
Right, and every receptor is tuned to a very specific modality. A mechanoreceptor won't respond to light, and a photoreceptor won't respond to pressure. This specificity is absolute. So when a stimulus hits a receptor, you get what's called a receptor potential, which is small and a graded electrical change. If enough of those change sums together, we can reach a threshold and generate a full action potential.
And the nervous system pays attention to change. Adaptation is key here.
Exactly. So some receptors will adapt quickly, like smell. You can walk into a room and notice an odor, and 30 seconds later it's gone. Or others adapt slowly, like proprioceptors, which we need constantly to inform us of where your limbs are.
I love the framework of modality, location, intensity, and duration, the 4 pieces of sensory information.
Yes, so for location, the concept of receptive fields is huge. Areas like the fingertips have tiny receptive fields with many neurons, allowing for very fine 2-point discrimination. And other regions like your back have a large receptive field, and that has a lower resolution.
Which is why the sensory homunculus looks so bizarre, as it has huge hands and lips, because the cortex dedicates more neurons to areas with finer sensitivity.
Right, and then there's going to be your sensory projection, and then that's the brain's ability to identify where stimulus came from, even though everything is just a neuron firing and arriving in the bundles.
And before I forget, we have to touch on referred pain, because people find this fascinating.
Of course, it's a classic example of a heart attack pain. The sensory fibers from the heart are going to converge onto the same spinal tracts as fibers from the left arm and jaw. And that brain is going to misattribute the source, so it feels like your arm is hurting even though it's an issue related to the heart.
This is a great reminder that the brain is making its best guess with the wiring it has. Now, let's shift to taste. People think that the tongue has taste map zones, which isn't accurate.
Yes, that is not accurate. So all 5 primary tastes exist throughout the tongue. What changes are the palpitae. We have the filiform palpitae, which provide texture, fungiform, which are mushroom-shaped, found near the tip, folate, and then we have that are more active in infants. And lastly, we have the valet palpitae, which are found in the big V-shaped row in the back of your tongue, and those taste buds are going to sit at the base of these palpitae, not on the top.
A key point is that taste cells bind dissolved chemicals using ligand-gated channels. If enough local depolarization occurs, they send signals through the facial or glossopharyngeal nerve.
And that's what most people, when they call taste, they actually mean taste plus smell, texture, and temperature, which is why hot coffee or hot peppers are going to activate those neuroreceptors, and they're pain signals, not taste signals.
Smell is my favorite sense because the olfactory neurons are actually exposed to the environment. They are the only neurons directly integrating with the outside world.
Which is wild when you think about it. So these odor more molecules are going to enter the superior nasal cavity and dissolve in the mucus, which are going to bind to your olfactory hairs. And these hydrophilic molecules are going to dissolve immediately. And then the hydrophobic ones are going to latch on to odor-binding proteins.
And the moment those neurons fire, signals go straight to the limbic system. That's why the smell is tied so strongly to memory and emotion. As the hippocampus and amygdala gets first dibs.
Yes, so when you're sick, the mucosal layer is going to thicken, and these chemicals aren't going to diffuse easily, and that's why everything, including your smell, is needed.
Now let's talk about hearing, because people often forget its mechanical sense.
Yes, so those sound waves in the air are going to hit the tympanic membrane, and that's going to move the ossicles, which are the malleus, incus, and stapes. And the stapes is going to push on the oval window, and that's going to convert the air vibrations into fluid vibrations inside the cochlea.
And once fluid moves, the base layer membrane ossiculates and bends the hair cells in the organ of the corti.
Right, and so those inner hair cells are going to detect sound, and outer hair cells are going to be like tiny muscles, and they're going to contract, tuning the cochlea and adjust the sensitivity.
Not enough people know that pitch is determined by where along the basal membrane the vibration occurs. High frequencies occur near the oval window, and lower frequencies occur further towards the apex.
Yes, and that amplitude is going to be the vertical height of the sound wave, and that's going to determine the loudness because more inner hair cells are going to fire over a larger region.
Now, let's go on to balance, which is essentially the vestibular system acting as our built-in accelerometer.
Right, so we're going to have two major components, the maculae in the euchroal and the sacral that's going to detect linear acceleration and static head position. Now the crista ampullaris is in the semicircular canals, and they're going to detect rotational or angular acceleration. And then we have the ultrolithus, which is going to be sitting on the ophthalmic membrane, add anterior to that, and that's when you tilt your head or accelerate, they're going to lag behind or bend the stereocilia.
And the semicircular canal detects motion in the three planes, twisting, cartwheeling, and somersaulting.
Right, and so they're basically going to tell your brain which way you're rotating in these three-dimensional spaces.
And now let's finish with vision. Light enters through the cornea, moves through the pupil, through the lens, and finally hits the retina.
And the retina is incredible. The rods are going to handle low light, and the cones are going to handle color and resolution. And in the darkness, these photoreceptors are going to release a neurotransmitter that's going to suppress these bipolar cells. And when light hits them, they're going to release, and that stops downstream when the cells activate.
And only the ganglion cells generate action potentials. Their axons form the optic nerve.
Exactly. From the eye, these signals are going to travel to the optic chiasm, then to the thalamus, and then finally in the visual cortex. Because of this hemidesegmentation, or partial fiber crossing, everything from the left visual field is processed in the right brain and vice versa.
A fascinating detail is that the fovea has the highest density of cones, so when we want high resolution vision, we move our eyes to project the image onto that tiny spot. Danielle, this was fantastic. I love episodes where we get to map the entire sensory world, how we detect chemicals, vibrations, pressure, photons, motion, and how the nervous system integrates all of it.
Thank you, Andrew. Every sensation we have in the brain, interpreting chemical electrical signals, is how the system is so beautifully engineered.
And to the listeners, thank you for joining us today. As always, you can find resources related to today's discussion in the show notes. Until next time, thanks for listening to the Huberman Lab Podcast.